So just to start off, a little bit of background, I guess, about nerves and stimulating nerves with light. It turns out that if you go back in the literature, some of the initial work was done back in 1891, and there's a really nice paper in French from that time talking about how muscle fibers can be excited by light, right? Now, keep in mind, this was about 110 years after Galvani had discovered that you could activate a frog's leg, the leg of a dead frog, by applying electricity. So, to some extent, I suppose that closed the loop, that we realized that you could use electricity and light to influence muscle fibers. And muscle fibers are related to nerve fibers in the sense that they're excitable cells. These are biological cells that can be stimulated in various ways. So realizing that light can be used to stimulate nerves or muscle fibers creates some interesting opportunities. We know, of course, that light is an excellent way of having a non-contact interaction with materials, action at a distance. We understand that light interacts with the electronic states of matter, and so it makes sense that if we're talking about an electrical phenomenon of nerve activity, that light can somehow interact with that. And of course we can focus light to a small spot, so whereas if we use electrical currents to stimulate nerves, those currents will spread out through the tissue. If we use light, we can hopefully get a much more precise and targeted interaction. So since 1891, there's been a bit more work done, and there's quite a variety of ways now that people are trying to use light to stimulate nerves. I mentioned here optogenetics. Optogenetics is a fairly recent field. It started about 10 or 15 years ago, where people realized that they could genetically modify nerve cells and introduce light-sensitive ion channels that would make those nerve cells excitable by light. So the classic example given there is using channel rhodopsin to make the nerve cells in the retina optically sensitive. So we're not talking about the light receptors in the retina, we're talking about the nerves that convey signals from the light receptors to the brain. So you can see immediately that if you can make those nerve sensitive and the light receptors are not functioning, you've got a way of introducing artificial vision. Another way that's been developed is to use caged molecules. So these are molecules that can be released when a cage is exposed to light. And there's a few other photoactive molecules that people have developed where by applying light you can change the shape of a molecule, and that will open or close an ion channel. And ion channels, of course, then allow electrical currents to flow around or through a nerve cell. A third method is to use infrared lasers, and it turns out that using mid-infrared light you can have a direct interaction with nerve cells to stimulate nerve activity, and I'll talk a bit more about that. The typical laser parameters that we're talking about for that sort of process, 1870 nanometers would be quite typical, and reasonably long pulses. These are modulated lasers, not ultra-fast lasers. Just for interest, this is an interesting picture of a rat that's been genetically modified to have light-sensitive cells in its brain, and by turning that blue light on and off, you can modify the behavior of the rat. I think it runs in left counterclockwise circles when you turn the light on. So you can see it's a very interesting potential, but a long way from being used in humans. Here's another nice example, and I'll just run this movie for you while I talk. So this is a quail embryo, so a baby quail, a baby bird, and there's the infrared light being delivered by an optical fiber here. This is the heart of the quail, the embryonic quail, and a laser Doppler vibrometer probe which is measuring any movement of the heart tissue. So what you're seeing down here is pulses of light and the pumping of the heart in response to that. And I'll just play that again for a moment. And you can see how there's a natural heart rate, but then once the laser starts to pulse, the heart rate is synchronized to the laser rate. So again, this is quite a nice demonstration from a paper in Nature Photonics a few years ago that muscle fibers or nerve fibers, we're not sure in this case, can be stimulated by light. So how does this infrared nerve stimulation work? One of the best indications we've had is from this paper in 2012. And what those researchers were able to show is that, well, very clearly, was that the light is absorbed by water. And I've put the absorption spectrum of water here. So 1870 is somewhere in this region. 1400 has also been used, and 2000 or so nanometers. So these are wavelengths that show us a range of absorption of water that can be used to have this stimulating effect on nerve cells. What they also showed in that paper is that the absorbed light is converted to heat, and that heat then changes the distribution of ions around the cell membrane. Cell membranes are polarized, so they create layers of opposing charges around them. And as you change the temperature, those charges will shift slightly. That changes the capacitance of the cell, and that can then lead to the triggering of voltage-sensitive ion channels. Some nice elementary electronics there for you. Another mechanism that was reported also in 2012 suggests that some other ion channels can also be involved in this process. So for those of you that aren't that familiar with nerve cells, the flow of charge is regulated by ion channels, and there's a whole lot of different sorts of ion channels. Some are voltage sensitive, but there's also others that are temperature sensitive or sensitive to particular chemicals. In this case, they were able to show that it's a particular heat-sensitive ion channel that seems to respond to the laser pulse here. So while the laser's on, you can see how there's that initial shift due to that capacitive effect that I mentioned previously. And then over time, the heat-sensitive ion channels open up until you cross a threshold, and then you get an action potential occurring. So they refer to that as a light-evoked voltage variation on this axis here. So the picture that emerges is that, yes, we can stimulate nerve cells with infrared light. It's water absorption that causes heating, and then there's voltage and, well, there's a capacitive response and then an associated response in heat-sensitive ion channels. From our point of view, though, well, so just to wrap up this discussion of infrared neural stimulation, there's a number of advantages that has compared to the other techniques like optogenetics and caged molecules, photoactivable molecules. It's relatively non-invasive, and we're comparing that with something like optogenetics, where you have to genetically modify the cells. But you can also compare it with traditional electrical methods of stimulating cells, where you need to place electrodes on the nerve cells, and that can create an inflammatory response and actually damage the tissue over time. Another advantage, because it's not an electrical technique, you don't get any electrical interference when you're trying to monitor the nerve cell activity. So to measure the response of the nerve, you still use electrical methods, but if you're using an electrical stimulation, that will interfere with what you're trying to measure. So these optical methods are quite attractive from that fundamental point of view of avoiding artifacts. And good spatial selectivity, because in principle you can focus the light down to single cells. But it's not ideal. Because we are relying on water absorption, and there's water in all biological tissue, it implies that it's very difficult to get the light through tissue to buried nerve layers. In the eye, it turns out that that's a major problem. You know, the front part of the eye is filled with the aqueous humor, and so if you're using these wavelengths to try and stimulate the retina, you can't transmit the light to the back of the eye in the first place. We also worry about the long-term effects of tissue heating. As you know, if biological tissues are raised in temperature by more than a couple of degrees for any length of time, they tend to get damaged. That's why we don't like high fevers. And we've also shown in this paper published a couple of years ago that if you try to have a number of stimulation sites and you have multiple pulses to convey information to the nervous system, those heating effects rapidly rise. The more sites you try to heat, the more total heating you get. So that's a limitation as well. So this is just really to summarize some of that discussion. We've got the infrared nerve stimulation up here at fairly high radiant exposures. There's been a bit of discussion about why the cochlea, the hearing organ, responds at lower power levels, but it seems as if it's more of an acoustic phenomenon. And again, that's some research that we've recently published. And then optogenetics is down here with much more attractive power levels, but with the issues around genetic modification and so forth, which are quite an ethical barrier for using that in humans. So what we asked ourselves is, can we make this photothermal process more efficient? And the idea we had was to try and improve the absorption of the light by using gold nanoparticles. So here I'm showing some gold nanorods, and many of you will be familiar with this kind of absorption spectrum that you get for gold nanorods, where the longitudinal plasmon resonance is here in the near infrared. That can be tuned depending on the aspect ratio of the gold nanorods. But that sort of wavelength turns out to be very attractive. If I go back to this graph here, you can see how if we rely on water absorption, we have to operate in this region where you're getting that problem of absorption in any other tissue. But if you can move down to this near infrared region here, water absorption drops off, and so you're getting much better transmission through the rest of the tissue. So now the challenge would be, can we associate the gold nanorods with the nerve cells to get localized heating? So to start off with, what we did is we took what's known as a cell line, and a cell line, I have a physics background, so it's taken me a while to understand some of the things that go on in biology. A cell line is an immortalized family of cells. And what this allows you to do, it's kind of like your laboratory standard of a response. With a cell line, you effectively are able to clone cells or grow cells that are clones of each other, and that allows you to do much more repeatable experiments. If you use naturally harvested cells, there's a lot of variability between the cells, and that of course means that there's a lot of variability in your results. So the cell line that we used in this work is a cross between a mouse blastoma and a rat glioma. So it's essentially a type of cancer cell, and being a cancer cell, it can be grown through successive generations without much change in the character or the nature of the cell. So we worked with that. The key thing, though, is that despite being a cancer cell, in this case, the characteristics that make it useful as a nerve cell model are still maintained. And there's an image here of one of the cells. You can see the cell body here with the nucleus in the process of splitting. And this part here and up there are the neurites. Those are the axons which carry the electrical signals between nerve cells. So what we did with these NG10815s was we incubated them with a solution of nanorods. In other words, we just added that to the growth solution of the cells, and it turns out that the cells under those conditions just take up the nanoparticles. They have a way of pulling them in through the cell membrane, and they become internalized in the cell. So you can see that here where we've added some rhodium dye to the nanorods, and you can see how they're glowing there inside the cell where they've been internalized. So the first thing we need to do is confirm that the cells aren't being adversely affected by these nanorods. This is quite a nice example again, because you can see the cell is in the process of division, and that implies that it's still functioning quite happily despite all these nanorods that it's taken up. But more interestingly than that, we found that the average length of the neurites, these extensions of the cell, actually increased once we added the gold nanorods and exposed them to light. So if you look at the control data here, where we only exposing to light, the average length of the neurites is fairly constant. But if we expose them to the laser with the gold nanorods, they actually increase in length. So it seems as if they don't mind or are even quite stimulated by having the gold nanoparticles inside. Some other results. The number of cells with neurites, the number of cells that were expressing neurites increased with both laser exposure and the particles, and the number of neurites per neuron increased as well. In this case, it didn't matter whether it was just the laser exposure or the gold nanorods plus laser exposure. Both of those factors increased the size and number of neurites. So from that, we actually concluded that the cells are quite happy to be exposed to the gold nanorods, and you could even speculate that this would be a good way of improving nerve therapies where you're trying to develop ways of improving damaged nerves, say from an accident, a spinal cord injury, for example. The next step, we wanted to see what would happen to calcium concentrations inside the cells. And it's quite well known that the signaling inside the cell that generates action potentials or generates the electrical signaling of the cell is related to calcium release inside the cell body. So we can monitor that using another type of fluorescent dye. We use a laser scanning confocal microscope to monitor that dye level, the level of fluorescence. And just using a 780 nanometer laser diode to stimulate to heat up the gold nanorods to match that absorption peak of the gold nanorods. the fluo 4 dye is excited in the blue, so there's a good separation between those two wavelengths. Now, it turned out to be a fairly tricky experiment to get a good synchronization between the scanning of the images and the pulsing of the laser. So there are some artifacts in this data, but by and large, you can see how, in this case, for example, there's some very clear spikes in the fluorescence level as we turn on the laser, indicated by these vertical lines. In some cases, the spikes in the fluorescence level were really quite significant. And we played around with quite a range of different parameters in terms of radiant exposure and pulse length. So just to understand what these spikes in fluorescence indicate, we've measured the intensity of the green light emitted by that calcium-sensitive dye, and we're able to see when does that intensity rise or drop, and plotted on this axis here. So we were initially a bit confused by that data because there's really quite a few different things going on. You know, for example, we're getting this quite strong response for relatively low radiant exposure. And when we go to a much higher radiant exposure, we're seeing a much weaker response. So we try to plot that out. Here's the radiant exposure, and here's the peak amplitude that we were able to detect in terms of those calcium spikes. And you can see how it does tend to drop off at higher radiant exposure. And when we looked into the literature on these sort of things, we realized that this is a quite well-known effect, that if you heat nerve cells too much, you tend to inhibit activity. So it's a bit like Goldilocks and the three bears. You know, the porridge has to be just right to get a good response. If we have just the right amount of radiant exposure, we get a strong response, but too little or too much, and it's not as effective. So that was a very encouraging initial result to show us that firstly the cells don't mind being exposed to the nanorods, and secondly there is this calcium activity provoked by the laser exposure. But we needed a more direct proof that there was electrical activity occurring as well. So the technique that we used for that is known as patch clamping, and I'll just try and explain that quickly with these figures. This is a fairly standard microscope giving differential contrast images, and we're able to bring an optical fiber in from the side to deliver the 780 nanometer light. And then from the other side, we bring in a very sharp pipette. So this is a hollow tube that's drawn down to a micron-sized aperture, and with that pipette, we're then able to contact the cell here. This is now a spiral ganglion neuron that's been harvested from a rat. And you can see the spiral ganglion neuron is about 10 microns in diameter, but with this micron-sized pipette, we're able to connect that to the cell, and if you're very careful, you can establish an electrical contact with the inside of the cell. So that's why it's called a patch clamp. You're patching it onto the cell. So what that allows you to do, if you've got an electrode inside the glass tube and another one out here in the solution, is you can measure currents through the cell membrane. Okay? The glass is an insulator, so the only path for current to flow is through the cell, out the membrane and into the solution. And at the same time, we can deliver the stimulating light from this other fiber. And just to compare, this shadow here is the shadow cast by the optical fiber that's delivering the light from the other side. It's a single mode fiber. So with that setup, we did a number of measurements, and this is just some initial work before we started using the gold nanorods. Here we are using the infrared wavelengths, 1870. We're using fairly high pulse energies, and this is just the potential that we apply to the cells as we do these measurements. And you can see how we get quite a clear but brief current flowing around the cell as we apply the laser pulses. The laser pulses are indicated by these bars. And the way we interpreted these pulses is that we were seeing that capacitive effect that I mentioned earlier. The heating, this wavelength is absorbed by the water around the cell. That heating effect then causes some current to flow due to the charge diffusion. Here we plotted the potential, and again you can see how during the pulse there's this change of potential, but then as the cell cools down, it returns to the baseline. So really just confirming that yes, we were able to see that capacitive effect. But at no stage with this experiment were we able to get action potentials. We never saw the very sharp, strong spiking of electrical activity that would indicate an action potential of being provoked in the cell. So from there we moved on to the gold nanorods, and there's some nice images here where you can see the gold nanorods inside the cells. These are dark field images. These are actually gold nanospheres that we used as a control. So the nanorods absorbing at about 780. The spheres absorb in the green at about, well, it's shifted to about 550 nanometers there. So that confirms that, again, the nanoparticles have been taken up by the cells. Then we exposed the cells to, this time to the 780 nanometer light, and also to a green wavelength as a control. So this sequence of images here is showing the response to 780 nanometers in cells that had gold nanorods, right? And you can see a quite strong current response to that laser exposure. Here we had gold nanospheres, and we exposed those to 780 nanometer light, and you can see a very small response in that case. So you really need to match the absorption of the particles to the wavelength to see this response. That confirms that we are getting a plasmon-mediated heating. And if there's no particles at all, then there's no response to the 780 nanometer wavelength. But what was really pleasing with all of that is shown here, where we've taken different pulse lengths of the 780 nanometer laser, and you can see how for short pulses we just get a brief voltage response, that capacitive response that we would normally expect. But then for the longer pulses, that sort of shoots up and we get this very characteristic action potential. So that's really what you're looking for if you're trying to stimulate a nerve cell, the action potential, which shows that the cell has fired and is sending a signal out. We also played around with some longer pulses, and we could get multiple action potentials during the longer pulses. So that turns out to have been the first data published showing that, or proving that you can use something like a gold nanorod absorber to enhance this infrared stimulation of nerve cells. And since then, we published that in just last year, but it's such a hot field, I think there's already 3 other papers come out confirming this result. So it's a nice example for students that working hard and getting the result out first is an important thing. In terms of where we want to go from there, one of the things that we felt a bit of a lack was the ability to measure the actual temperature. So yes, it's easy to talk about how this is a photothermal heating effect, but you really need to be able to measure the temperature to confirm that that's the case. We did some work a few years ago where we tried to use fluorescent dyes again to measure temperature. Turns out that rhodium B is temperature dependent, and you can see how here we were able to use the lifetime, for example, the fluorescence lifetime to measure temperature in a nice range around 37 degrees around body temperature. But it turns out that as soon as you put that dye into a biological medium, a biological tissue, there's all sorts of other factors that start to interfere with the fluorescence, pH, and I guess some other chemical components inside the cell start to interfere with the temperature sensitivity, and so you lose the ability to measure temperature with that. So what we realized is that you can use the same patch clamp technique that we use to measure the electrical response to measure temperature. And I'll just quickly go back to this picture. Basically what happens is, because we're measuring currents that flow through the open end of the pipette and through the cell membrane into the solution, one of the limiting factors for that current is the size of the aperture here. And obviously if you're getting heating in that environment, that opens up the aperture, decreases the resistance, and so you can start to use that as a temperature measurement. So using the patch clamp in that way, we were able to again apply these different length pulses and see how we were getting different levels of heating. And it turns out that these kind of temperature changes are also quite typical of what people have seen for infrared neural stimulation without gold nanoparticles. So, in conclusion, I think there's really quite a few exciting things that we've been able to demonstrate with this work. We've got all the positive aspects of using light to stimulate nerves, that it's non-contact, that it can be focused to small spots, that we reduce inflammatory responses and so forth. And using the nanoparticles, we're able to decrease the power levels to a more comfortable range. And this really does make a difference. When we were working with the mid-infrared wavelength, 1870 nanometers, we were using some very powerful lasers, and it was much easier to kill the cells than to get any response out of them. Here we're using a much lower power laser diode, 780 nanometers. There's lots of cheap laser diodes around, and so it's a much more convenient wavelength to work with. And of course, very importantly, we're in that transmission window of the tissue, so we can start to think about reaching deeper nerve cells inside the body. Of course, I'm not trying to pretend that we really understand how this process works. Yes, we believe that there's some capacitive effect. We believe that we might be triggering these temperature-sensitive ion channels, but there's more work that needs to be done to confirm those theories. And based on what we've seen, we're really excited about the potential for new types of neural prostheses based on this approach. And I'll explain that a bit more in the next slide. So as I say, there's certainly a lot more work to be done. to understand the mechanisms. We're still puzzled about why experiments that are done inside a living animal in vivo are often quite different to experiments that are done in vitro, so with cells in a glass dish. So that's a question we'd like to answer as well. And I think most importantly, if we're going to start injecting nanoparticles into tissue, it's well known that nanoparticles will just tend to wash through the tissue and they end up in the liver. We don't know what they do there, but that's where they end up. So really we'd need a way of getting them to stick to the nerve cells and stick there for long enough to have some benefits in terms of stimulating the nerves. So in terms of prosthetic devices, what could we imagine? And this is really, I think, what drives the work. I mean, it's great fun playing around with lasers and cells and making them respond. But why do we really do that? Well, I believe that we might be able to help the blind to see, which is quite an exciting challenge. So there's a lot of work going on at the moment with bionic eyes, but relying on electrical stimulation to generate activity in the retina. Here's a picture of a cross-section taken through the retina. This is, I think, using optical coherence tomography, and it's quite an amazing image. This thinner region is what's called the fovea. It's the sensitive central part of your vision that you use to recognize faces and to read. And it's got the highest density of receptors in the retina. So the density of ganglion cells in that region is about 40,000 cells per square millimeter around that region. Now, 40,000 per square millimeter, when you look at the density of electrodes on the current retinal implants, they might have, I don't know, 10 or so electrodes in a square millimeter. You use 10 to stimulate 40,000 cells. Well, what's that going to do? There's a bit of a simulation here which shows what's likely. Here's a face. We sample that face at a resolution of, I think that's something like 100 pixels. But then if we decrease that resolution to the level that a retinal implant can achieve, you end up with something like that. So the reality is that, yes, you can put these implants into the eye, but because the current spreads out and there's so few electrodes that you can build into these implants, you end up with pretty useless image. So I'm not saying that these implants won't be used. For example, what this shows you is you won't be able to recognize faces with these implants, but it might be possible to use these kind of patterns to read. So you can easily imagine a vision system that would detect text and convert text into a kind of visual braille pattern that would be able to allow a person to read much more quickly than you can read by touching a braille pattern. So there will be some benefits to these kind of implants, but it's just impossible, physically impossible to get the kind of resolution that would start to be a meaningful version of vision. And it relates to the way that the current spreads out through the tissue and how much current you can inject into the tissue before you start damaging it. So yes, we can make much smaller electrodes, but you can't use them inside the body. So that was 1,000 pixels there. So I think you can start to see that the only way I can imagine that you can start to stimulate individual nerve cells in that region is by using light. And if we can find ways of injecting gold nanoparticles into the space near the retina and get those nanoparticles to stick to the nerve cells, I think we've got a good chance of getting at least as good a quality image as that. So just a few acknowledgements there. There's been 4 PhD students involved in progressing the work to where it is today. And it's one of the nice things about working in this biophysics area is you get some nice images to put on journal covers. Just before I finish up, I've just got a couple of minutes. So you might have noticed in the title slide that I'm director of a training center in biodevices. So I thought I'd just say a few words about what that is. There's the website there if you're interested for some more information. What we're trying to do in Australia at the moment is to modify the way that PhD training is taking place. So traditionally, and I'm sure it's a somewhat similar situation here in Lithuania, traditionally a PhD is a very academic qualification. Essentially, you learn how to be an academic, how to do research and how to publish and communicate your research. And those are very important skills. They are skills that can certainly be useful in high-tech industries, but I think in Australia we've had such a strong emphasis on that academic side that industry hasn't really valued PhD graduates. I was talking to a company in Melbourne, and they said, the CEO said to me, yes, we do hire PhDs, but it takes a year or two to get them up to speed. Okay? And if you think of how long you've spent at university by that time, that's not a good indication of the skills that you have. So what can we do about that? So the PhD that we're implementing in this training center is partnering with industry from start to finish. And what that means is that the academics aren't proposing the project. The students are talking to academics, to the industry partners, and then to end users of medical devices to understand what their needs are and then to propose the project. So what we end up with is students that pitch a project as if it's a startup. They pitch a project to the industry partners who then select the project. And we have a big focus on planning the research and delivering the research on time. So, using industry discipline to try and do things in a timely way and keep on budget and so forth. So to support that, we've started a new PhD degree, which we call PhD in Technology Innovation. And we do some really interesting things to try and come up with some really innovative solutions to the problems that the students identify. One of the keys that we've understood is that to come up with innovative ideas, it's best to be working in a group. So the first part of the PhD is actually team-based rather than individual. And we also take some coursework units from our Masters of Entrepreneurship and Innovation. So that's just a very brief summary of that training program, and if anyone's interested in that, I'm more than happy to talk to you later on. So I think with that, we've got a few minutes for questions, hopefully.
Yes, thank you. So we have to have questions. You said that most of the phenomena you are observing are possibly based on temperature changes near a membrane. Mm. And so my first question is, the capacitance changes you suggested at the membrane, you said there may be a diffusive effect of ions on one side or so, and on both sides. Could it be also that the membrane changes in thickness? And that's a very simple explanation for a capacitance change.
Absolutely.
Is that been observed?
So you make a very good point. We know from the way that capacitance is defined that the spacing of the plates determines the capacitance. So, you know, that was looked into by that Shapiro paper that I mentioned, and essentially what they rely on the modelling of the stern layer and the whole membrane system as an equivalent electrical circuit to argue that thickness change isn't enough to explain what they see. So, you know, they know the size of the membrane and they know the thermal expansion coefficient. So based on modelling, they argue that, yes, if you put those parameters in, you get a small effect. But to explain the larger effect they observe, you need to have some other process. And so it looks as if the size of the effect is dependent on the double layer changing its size as well.
But I understand the diffusive layer is always small, thin, if you have a high ionic strength, which is the case in a cytoplasma or whatever. Mm-hmm. So isn't that a contradiction?
Well, I think the point is this, that the thermal diffusion can spread that double layer out more than the thermal expansion of just the membrane.
A second question about your vehicle, you know, your gold rods you use for heating. But of course the gold rods, they're coupled resonantly with your laser light, and so you will have to expect also at the tips a strong plasma, surface plasma, and that may heat the water. But if it's in the near field of any structure in the membrane of a protein, and so they may affect really strongly, maybe even destroy locally, because you have a strong near field effect. What is your thinking about that, instead of just heating gold?
Yeah, so you're right. There's two processes there, and we're assuming that the dominant process is that the plasmon resonance, you know, that energy is rapidly transferred to the lattice of the particle as heat, and that then heats up the surrounding environment. But what about the near field? I'd have to say we don't know. And I do a lot of surface-enhanced Raman scattering as well, so I do think about these things as well. What I'd like to do in the future is to do some more careful experiments. And for example, you know, tip-enhanced Raman scattering. If we can start to scan over the cell using a tip-enhanced Raman scattering probe, we might be able to see, are there hotspots? You know, can we find an association between the tips of the rods and particular ion channels that are responding? So, you know.
That's the experiment you should do. You have one single rod.
Yeah, and in fact, so exactly, the tip itself can become a source of near-field excitation. Yeah.
More questions?
That's not good. I must have confused everyone.
Okay, I can try one. When we talk about light, then there is the intensity of the light, and then there is the energy of the light. And which one of them are inducing these neurons? The energy, or
that's a good question. So going back to that plasmon interaction, it's the electric field, right? So it's not the intensity, it's the electric field that is exerting a force on the surface electrons in the metal particle. And so there's a movement of charge on the surface of the particle that creates a dipole, an oscillating dipole. And you know, that's confined to the surface, but of course as those electrons are pushed backwards and forwards by the electric field of the light, there's collisions between the electrons and the lattice, and that's what generates the heat, that sort of collisional decay of the excitation.